Okay, I hope people can hear me. We're going to be giving you a little rundown of the exciting field of protein design for all you iGEMers today. Because I think it might be of use to your team and some of your future iterations. So just as a brief structure for what we do, I'm just going to give you a little introduction about kind of the background of protein design and why you might want to care about it. And some cool things it's done. And then I'm going to pass you on to, that's written the wrong way around, isn't it? It's true. Slides. So are we doing you then Felix, or should we do, I think we should do Felix first. So then I'm going to pass you over to Felix, who's going to tell you about his research into developing de novo photosynthetic proteins, which is really cool, some pretty colors. And then we're going to have a bit of opportunity for questions. Then passing you over to Thomas, who's going to walk you through a redesign protocol of a natural protein that we're working on. Sheffield's iGEM team are working on currently. With a bit of time for questions. And then we have our very own famous Professor Williamson, who's going to be talking about maybe some of the limitations of these AI tools and how far can we take them. So there will be a point in your project where I think you're going to reach nature's limits. I think you'll need a higher fidelity of engineering. I think we're getting to a point where simply shoving bits of protein together and hoping for the best, although it's had some very good results over the past 20 years, is kind of coming to an end, and you will need a more precise way of engineering the bits to make sure that they don't fall apart. Remember, the parts of proteins that you are sticking together are only within evolution's purview, and therefore they haven't evolved to be stuck together. So you might need to be a bit cleverer and design an interface between them to make them stable. But there's a big problem in biology, and aside from that being something that all my supervisors have said to me, there's also the protein folding problem, which is how can we design proteins when we're not exactly sure of the exact mechanisms of folding? And this is kind of embodied by this here, which is known as the Levenfeld paradox. If you have a 100 amino acid protein, there's some predictions that the number of possible combinations due to the flexibility of, the orientations of different peptide bonds, you can theoretically adopt up to 3 to the power of 198 different conformations. So how can we predict from the vast array of conformations which one is the native fold? And it turns out that a clever way to do this is to leverage our computational prowess, so that this search we can kind of perform in silico and hone in on the native fold. So if you look on the, can't tell on the left and the right, on the left-hand side of that diagram, that's kind of the protein folding process. We can input a primary sequence and programs such as Rosetta and AlphaFold, which are trained on large amounts of data from the protein data bank, which obviously does have links between sequence and structure. You can put in a new sequence and it is able to predict, based on this training, what it will fold to. And then from there, a bunch of clever people figured out that you can invert these programs and say, okay, if I have a 3D shape that I want to design, give me a sequence that will fold to it. And by using very similar architecture, you can do that, which is the field of protein design. And whereas kind of past attempts used random sampling and energy minimization to do this, more and more we're using artificial intelligence. And yes, so programs such as AlphaFold have a design component. Rosetta have recently published their all-atom design feature using a diffusion network, which is really cool. So more and more protein design is becoming easier leveraging artificial intelligence. Now, design can involve everything from taking a native structure or structures from natural proteins and using the design software to tweak them in a way that maintains or even enhances their stability, and that's kind of called protein redesign. But you can go completely the opposite way, the other end of the spectrum, and go completely from scratch, develop a novel shape from scratch and a sequence that follows it. And that's what we call de novo folding. Yes, next slide, please. So there's been some really cool examples of how this has kind of pioneered lots of little facets of biology. It's been used to make a bunch of cool therapeutics and vaccine candidates. Pictured there is a lab designed some inhibitors for the one of the influenza entry proteins. So they basically modelled exactly where they wanted to bind it, developed a complementary set of residues that would bind it.
I can see your hands.
Hi, Owl. So yes, the yellow bits on that diagram are the kind of residues where they wanted to bind. They said what residues would bind these, and then told a design program to design a scaffold that held them in the right place. That was one of the things we do. It's big in medicine at the moment. We've got proteins that form Boolean logic, true, false logic. They can land on a cell and make a kind of calculation based on what antigens are at the cell surface, whether it should open and deliver a therapeutic or a cell signaling molecule. That's called locker. It's a really interesting one. You can design multiple modes of symmetry in protein fragments such that you can self-assemble them in vivo, spontaneously, and even attach specific binding sites that you can assemble, like a buckyball that has antibodies sticking out of it to bind whatever you want. And we've even been able to incorporate small molecules and ligands, which is really important in developing new photosystems. So this is from a very recent paper, co-published with some people in Sheffield even, where they designed some new scaffolds to bind chlorophyll. So just to finish off my introduction, there are definitely two facets to this kind of design world. Protein redesign, which is what it may be more immediately applicable to current nitrogen teams. Maybe if you've got a construct that you've fused together, but it's not quite surviving in vivo, or it's not quite working as well as it could. Redesign can take that existing fold and maybe tweak it to make it more stable, maybe more tolerant to a range of pHs, or maybe shift its binding specificity. But that involves taking something that already exists. If you wanted to be a lot more adventurous, maybe you had an idea for a protein that didn't exist, you wanted to find a molecule for which protein doesn't exist for it yet. Maybe you can get into the mysterious waters of fold de novo design when you can develop a brand new fold. And I think a mix of those two schools of thought is going to be told by, we're going to go to Felix now. So Felix is going to show you exactly what he's been tinkering with, which is actually probably quite reductive, because it's actually much cooler than just tinkering. Yeah, so enjoy his celebrating friends and other stories.
Cool. So, yeah, as I mentioned, I'm talking about some de novo billet proteins. So I'll just quickly go into a bit of background about what billins are and stuff, so that the project makes sense. But billins are sort of universally found in all organisms as a breakdown product of heme. But you can kind of see they've got these in a macrocycle. There's quite a constrained conformation, but once you break the ring open, they lose this constraint, and that results in kind of changes in spectral properties. So you can imagine if you've got a molecule flopping around in solution, it can occupy multiple different conformations, and this can change the sort of electrostatics within the conjugated pi system, and this results in this sort of can everyone see the mouse?
Okay.
Get that laser. Nice, yeah. So when you're kind of unbound in a protein, you've got this really broad absorption peak. And that's basically because there's multiple different conformations, all absorbing at slightly different wavelengths. But if you put them into a protein and fix their conformation around a certain range, you get this real increase in the absorption signal, reduction in the full width at half maximum, and basically, because you can, so the area under the graphs of these two peaks should be effectively the same, but now you've taken a bunch of these species that are like hiding out on the fringes and put them all into one species. So that's why you get this sort of enhancement. And this is what the billins look like when they're in proteins. This is fibrobillin, flexinobilin. But the other interesting thing that actually happens when you constrain the number of conformations these molecules can take is the pathways for de-excitation once you've absorbed the photon, they reduce. So normally you can imagine it's flopping around in solution, it'll absorb the photon, and it can dissipate that energy kinetically by wobbling around. But when you can constrain it in a protein, it's no longer capable of doing that. So the pathway to release this energy by fluorescence increases to the point that a natural light harvesting protein with one of these billions in has a quantum yield close to 1, which means that like almost every photon that it absorbs re-emits as another photon, and that's obviously very important for light harvesting and photosynthesis because you want to get the most out of the photons you absorb. So these billiproteins are found in these humongous megadalton structures called phycobilisomes, and they sit on top of photosystems. And you effectively have an energy cascade from the outside to the inside. So the spatially distant bilin proteins will absorb further to the blue, and the ones that are spatially located close to the reaction center will absorb close to the red and will have an overlap with the reaction center. And in this way, you can kind of cover a large portion of the spectrum. So this can be seen in, there are lots of different types of photosynthetic organisms. There's cyanobacteria, there's eukaryotic phototrophs, which most people are familiar with, like plants. There's also purple bacteria. But in photosynthetic eukaryotes and in purple bacteria, there are these gaps in the wavelengths they absorb. And in cyanos, they actually cover this gap by using phycobilisomes. So you can see how they can actually enhance the amount of energy you could pass into a reaction center if you don't have this already. And they basically represent a really interesting target for trying to span a range of different wavelengths by using slightly different soluble proteins that all absorb light in different ways. So, as I mentioned before, bilins are almost found in all organisms universally as a breakdown product of heme. But some of these photosynthetic organisms, mainly cyanobacteria and red algae, have co-opted them to do with light harvesting. And so there are multiple different bilins that exist out in nature. But one I'm going to focus on today is phycoerythrobilin, and it's made through the breakdown of heme by heme oxygenase into biliverdin, and then turned into phycoerythrobilin by a synthase enzyme. And a lot of these light harvesting billiproteins, they bind the bilin by a thioether linker to a cysteine, and that needs to be recognized by a motif, which is the CXRD motif highlighted here. So this is a sort of alignment of lots of different organisms, phycobilisomes. And a molecule called a lyase, CPCF lyase, takes the bilin once it's been synthesized, moves up to your billiprotein that's not got bilin on it, orients itself along this CXRD motif so that the correct portion of the bilin is facing towards the cysteine, and it allows the thioether bond to be formed. So we were wanting to make some de novo billiproteins, partly with the idea of you can make lots of different proteins with one bilin molecule that absorb light in various different areas. So this would be an interesting way to introduce de novo antenna proteins to a new organism, and we can tune the spectra that those proteins absorb at. And then you can start stacking them together and you get a funnel that would be like that of a phycobilisome. So there are these two tools that Tom will probably be speaking about later, this RF diffusion and these two new MPNN tools. So diffusion can generate novel backbones either around a protein or around a small molecule. And what's interesting to me is doing it around a small molecule. I've got a small video showing how it can be used to design a protein binder just so you can get an idea. And the tool effectively creates a bunch of noise around the site that you're interested in, and then it slowly sort of packs them together in a way that that you can recognize as a protein backbone. And it does this by effectively denoising the area and the space, and then threading through a backbone in a way that it thinks would be sensible. So you can imagine where you've got a small molecule, you create a bunch of noise around that, and then the program thinks about, okay, if I've got a beta sheet here, is that going to be better buttressed up against my pigment, or is it going to be easier to do like a ring of alpha helices? So you can imagine this sort of thing happening with a small molecule as well. But the problem is it generates a backbone that doesn't have a sequence. And now the sequence needs the structure needs a sequence, and that's where MPNN comes in. MPNN is almost like an inverse folding tool. So AlphaFold, you give it a sequence and it gives you a structure. Protein MPNN gives you the probability of a sequence giving a structure. So you put in this like polyglycine backbone that has a specific topology, and then it will give you out new sequences that will fold into that topology. This is just a little bit pulled out from the protein MPNN paper, but Isaac mentioned the inversion of AlphaFold to make designs, and you kind of get this around 10 to the 1 mg per liter of a soluble protein. But with this MPNN tool, you get up to 10 to the 3 mg per liter. So that's a gram of protein per liter of culture, which is insane. And you see it across all designs using MPNN. And moreover, when they got these structures out and they saw the crystal structure, there was actual like rotamer agreement between the crystal structures and the MPNN model. So it's clearly a very useful tool. Okay, so we were working with the Baker Lab who developed these tools, and we came to them with our idea of making some sort of de novo billet proteins, and we gave them this the conformation of a bilin in a native protein called CPCA, and this small chunk of backbone, the CXRD motif, so they could be recognized by the layers. And they developed several different folds using the RF diffusion tool, and they're all Q's fold seek. You can't find anything in nature that looks like them. If you use BLAST, you can't find any sequences that look like them. So these are completely novel protein sequences. But the problem with this kind of stuff is, so we had to trial 94 designs, and we found about 10 that bound bilins and 3 that were really good at binding bilins. So it's a very high throughput problem at the moment, but the fact is we can do it. But what's interesting that came out of some of these designs is that we get quite a nice span of spectra. So this is the sort of wild type that it was almost based off, so at 557 nanometers. And then some other designs out at 573 and all the way out to 607 nanometers. So just using like a single molecule and just deciding to use different backbones, you can actually span a whole range of different possibly electrostatic environments or conformations within the protein. So that could be potentially interesting in the future for enzyme design, where you need to stabilize a particular transition state or something like that. Moreover, these proteins are so stable that when you run them in SDS, like a denaturing gel, and take a fluorescence spec, you can actually see the proteins fluorescing in the gel, because that means basically they probably haven't unfolded that much. And yeah, running sec on them, they kind of all look fairly single species, which means you haven't got loads of aggregation. And we have so these are all fluorescence emission and fluorescence yields with respect to the sort of wild type of designs based on. And we've got a range of Stokes shifts, so about 11 nanometer Stokes shifts for the 2 billet proteins absorbing in sort of 500 to 600 range. For those that were absorbing over 600, actually had a reduced Stokes shift, but this actually happens when you have more negatively charged environments, so it's somewhat to be expected, because actually these binding sites for the far redshifted ones do look very negative. But so something to note is the fluorescence yield, like our best one, we got like close to 60% of that CPCA, which when it first happened, I was like mildly disappointed. So I was like, oh, why didn't we beat them? We used an AI tool, but it's obviously nature's had billions of years to evolve, and they probably know what they're doing better. So for a first round of designs, it's pretty incredible to actually even get that close to the native protein. So as you saw before, those glycobilosomes are actually these humongous structures, and they pack loads of pigments into a sort of area, and that's their advantage. They kind of build into the sort of cytoplasmic space, whereas a lot of other pigment binding proteins sort of occupy the membrane space and try and pack as much pigment as they can in. But a glycobilosome extends itself out using all that cytoplasmic space, but just allowing you to pack more pigment per sort of reaction center. So we were hoping maybe we can try and recreate something like that.
And this might be something more interesting to a lot of you guys based on what Isaac was saying with trying to get two proteins to stick together. And you actually you have to test a lot less sequences to get these sorts of things to work. So this is almost on the redesign side, even though we're using de novo proteins, but it's kind of like we're taking them a step further. So this is an AlphaFold 2 model of C11, one of the original designs that came out of the screen. I'm trying to fold it as a trimer. And I don't know how familiar everyone is with AlphaFold, but you get these PAE scores, which is predicted alignment error. And within a box, it's like, how good is chain A folding with respect to chain A? But then the box to the side, this is chain B, this is chain C, and it's trying to tell you how well chain B and C are placed with respect to chain A. So obviously this is an alignment error, so low numbers is good, big numbers is bad. And you can see chain B and chain C are up in the 30s, suggesting that AlphaFold doesn't think it's placed them very well with respect to the other chains. So this means we probably haven't got any sort of oligomerization going on. And when you kind of look at the insides of the model, there's no obvious charge patterning, everything looks fairly negative, so it's going to be quite repulsive. So using a computational pipeline, which uses a tool called ESM fold, which if you're trying to fold lots of sequences, is recommended over AlphaFold because AlphaFold can take a long time to run. So I think for a given trimer, it took about 9 seconds to run one fold. So I was able to use ligand and PNN to generate 10,000 sequences and fold them all and decide which ones were following the best. So the idea was try and find an interface where a trimer might be happy to sit, because you can imagine for a given monomer topology, there are going to be different orientations you can stick the protein together, and some are going to be more favorable than others. So I allowed any of the sequences, any of the residues to change, and ran this ligand and PNN with all 3 monomers in the file. So it was trying to sort of bring them together. And I ran this 4 times until I started to get an agreement between ESM fold and AlphaFold of proteins that actually folded into like a nice distinct trimer with good low PAE. And once I found a sort of structure that formed a trimer, I went back to the beginning and only allowed the interface residues to change and repeated the cycle. And then I again filtered based on whether AlphaFold also predicted trimers. Because something you can get with different design tools is, because these are all trained models, there are like biases within the models. And if you only use one tool to do your design, then you might have what they call like adversarial sequences. That's the actual word that they use. But it just basically means that it's like an error within the model where it's like, actually it thinks that this is going to work when in reality it won't. So it's good to like cross-check between different models, because then if both models agree, then you can say, oh, I solved this with respect to both models, therefore it's probably true in reality. So I found 4 of my favorite sequences that came out of the sort of thing. They all AlphaFold very well. You remember the C11 fold when it was done as a trimer? All of these were red, but now they're all blue, which means we've got values for the predicted alignment error close to 0. And actually, when you sort of explode these trimers and look on the interfaces, you can see some sort of charge patterning going on, negatives to positives, and you can also see some sort of hydrophobic packing where all this the white surfaces are displayed. So I ordered all of these genes and transformed them into plasmids. This was so much easier work than doing it with 96 at the same time. But I'll just quickly go through the worst design and show you the little things I did to analyze whether I had gotten this new oligomeric state. And then I'll show you the best design. So this was the worst one. We can see that the absorption looks fairly similar to that of C11, which I showed earlier. DSDS page from the elution of this, I purified by IMAC, like with a his tag. It looks fairly clean. We've got loads of protein. Running it on native page, we can see there's quite a high oligomeric state. So when I ran it on size exclusion, we actually get this shift in the peak. And if you have something that elutes earlier on size exclusion, it means it's larger, it's spending less time in the pores on the column. And you can see C11 is this sort of orange peak here. and this is close to around 20 kilodaltons. So that's what I'd expect for the monomer. And then the trimer designs actually a loop closer to the void volume, which is around 60 kilodaltons. So that is where I'd expect the trimer to be. Running these things on native page, this just is like running an SDS gel, but you're not unfolding your protein. You're trying to maintain oligomeric states and doing a fluorescence scan. I actually found I had like 2 bands, one around 120 and one around 60. So that possibly suggests I've got like multiple different oligomeric states present. But you can see my bilin signal with respect to the protein signal is really quite poor, which means that either we're not constraining the bilin as well as we were previously, or we're not binding as much bilin. So you can see this in what you'd expect in a 1 molar protein solution. The absorbance for this new design is about 1,000, but for C11 it's up to 7,000. So then this is the other design that kind of came up quite well. This really cutely named 7754. You can already see from the elution from the iMac it's quite a lot darker. The absorption is fairly similar to that C11, and we've still got this peak that elutes earlier than the C11 peaks, just in we've got high oligomeric state. But the best thing to see was this value of the bilin signal in a 1 molar protein solution is close to 13,000. So that's almost double what we have for the original design, meaning we're either constraining the bilin more or we're binding more bilin. It's hard to tell at this point without structures and mass spec. So I took these things down for negative stain to see if, well, firstly, if we had nice monodispersed particles that all look fairly similar, and also to see if we could see any sort of hexameric states. And there were a couple of things. So this guy here, which is here on the negative stain image, it looks a little bit like 2 trimers stuck together. And these are just like models predicting what they might look like to see what they are. So yeah, I'll just leave it there. But all the takeaway is that there's novel methods for designing soluble proteins to bind ligands. And you can also create novel interfaces to generate your own oligomeric state, depending on what you want to do. Thank you. Yeah, thanks to everyone involved.
So, we're going to pause for some questions. I think it's just worth saying, that was awesome, by the way, that's so cool. That I think one of the most useful things that iGEM teams can take from this is just how literally off the bat using ligand MPNN, you can put a fold in and get a sequence that, an alternative sequence to that fold out that is orders of magnitude more soluble.
Yeah, exactly.
So if you've figured out that your task is solved by a structure that involves sticking 2 things together, but when you try and express those 2 things stuck together, not working, try feeding that shape into ligand MPNN, and it should spit out something that folds and expresses better. And once you've got that going, maybe you can make some other cool stuff. Do we have any questions?
There's no questions. If you want to put some questions in the chat.
Yeah, something else to say, like on what Isaac was just saying, if you have a protein that has a function and you want to improve it, like people from the Baker Lab have used just protein MPNN to change the sequence of the structure, express a couple of them, and then they test their turnover, and you can get better enzyme rates like Km over Kcat over Km.
Yeah.
And that's literally just with almost random sequence design, like sequence design maintaining the shape, but you just maintain your active site, and something about maybe the stability of the protein or the conformation of the protein is ever so slightly different, and it can favor a higher rate.
Yeah.
There's a question which says, why do these tools increase solubility?
So probably, I don't think anyone knows, but they're all trained on the PDB, and the PDB is based on things we've solved, and things we've solved tend to be more soluble because you can express them in E. coli or something. So it's probably a slight quirk of the training data set.
Yeah, I guess if they've been trained on proteins that are able to easily purify and very crystallize, then they're going to be biased towards structures that allow that to be done.
And something else is like a lot of the stuff in the PDB was expressed in E. coli. So a lot of these proteins express really well in E. coli, but not so well in other organisms. And that is probably, again, because they're trained on things that express well in E. coli.
That's
Can I
that's
ask
fine.
another question?
Yeah.
So from what you said, I got the impression this can be really quick.
Yeah,
So you basically have an idea, you feed it to MPN, you get answers back in 12 hours, and the next day you can design your plasmids.
yeah, exactly. It can be. The initial making of maybe a pipeline can take a little bit of tweaking sometimes, but once you've got something that's running well, then you can just set it off. Like the oligomer designs, I don't think once I found out an interface and once I had the pipeline, it didn't even take a day to run. And then I ordered the genes, and then beginning of next week was running sec.
Yeah.
Yeah.
Well, yeah, I did a master's project with Felix trying to make a crossover version, which was a lot more difficult as it turned out.
Well, this was also pre all of these new tools.
Yeah. Even then, when MPN was in its first stages of being released, I think my first task was redesigning one of your designs, and the sequence was spat out the same day, and we'd ordered the G-pod the same day.
Yeah, exactly.
So literally the first week of me being in the lab, by the end of that week, we'd had it at least expressed in E. coli. Pretty quick.
But the diffusion can take a bit longer. So I think in my hands, I find like 10% of the topologies that come out of diffusion are absolute crap. Like you put in a carotenoid and it goes like, here's an alpha helix, and then the rest of the protein. You're like, no, I want to do it here. And then what I found was like generating 100 designs and then sifting through almost by eye, like, yeah, that looks like it's a sensible protein. And then you pull those out, and that can take maybe 24 hours actual running and then sifting. But then once you've got like an actual topology, the protein MPNN, ligand MPNN runs so fast. And then if you're using ESN fold, you can fold 10,000 things in like 6 hours or something. Yeah.
It's worth pointing out that details of some of these tools and how they can be used are going to be written in our handy AI guide that will be published onto the IGM universe soon, within the next couple of weeks, I think.
So also the GitHubs for all of these tools. I don't have a computational background, and I just followed like the GitHub recommendations of install and running. And
They're very user friendly.
yeah, they really explain like how to do everything.
Any more questions?
No more questions.
Shall we
Thanks.
jump forward to, yes, thank you, Felix. Shall we jump forward to Thomas, who's going to give us, hello, sorry, I keep forgetting where I'm looking. Thomas is going to give us an explanation exactly how you can practically use these tools in the context of our IGEM project, in which we are redesigning a phosphatase that doesn't undergo end product inhibition, but it's very useful for sequestering phosphate from the environment to counter the depleting reserves we have. So Thomas is going to talk a little bit about this enzyme and what he's been doing to redesign it.
Yeah. Hello. Well, I'm going to go through a walkthrough on how to basically do the protein redesign stuff. So how we use, well, I'll go through it. So for this walkthrough, for this demonstration, we're going to be using ligand MPNN, Chimera X, Alfold server, and conserve. And then, so ligand MPNN is our protein redesign tool. So that'll basically, we'll pass in the structure, it'll pass our sequences, and then the sequences should be improved. Chimera X is just for visualization of our structures. AlphaFold is for getting the structure from the sequence. And then conserve is used to find the best residues. So this will be a section that I'll go into more detail later on, but this is for finding residues to fix with ligand MPNN. So then we can try and keep the functionality of the protein that we want to be redesigning. But there's plenty of other tools to use. So there's protein MPNN, which is basically, it's the same as ligand MPNN is the same as protein MPNN, except you specify that you want to do it, including ligands. There's docking tools available like CVDock2 and MIB as examples. So CVDock2 is for docking ligands into proteins. MIB is for docking metallic ions into your protein. And then there's different visualization tools as well. If you prefer to use IMOL instead of Chimera X. So there's plenty of tools, but for this demonstration, we'll be using ligand MPNN, Chimera X, AlphaFold server, conserve. I would say it's best to download ligand MPNN. You'll have to download ligand MPNN and Chimera X. So the website for these two. So to download Chimera X, I advise downloading 1.6 because at the moment there's a tool which is only available for 1.6 called iSolder, which basically lets you identify different cofactors which you're going to put into your protein. So all you'd have to do is go into this website here, and then you can download Chimera X, and it's pretty simple. It's just an installer. And then for ligand MPNN, you've got the Darkroast ligand MPNN, and all you have to do is copy the repository to your computer, and then follow through the commands that are available here. So back to the walkthrough. So I'll go through the general overview of what we'll have to do first. So first of all, we'd have we've got the protein, the sequence of the protein that we want to redesign. So we'll start off. It's not in the cycle at the moment, but we'll start off with our sequence that we want to redesign. We first want to get the structure of our sequence. So for that section, we'll be using AlphaFold. We'll then want to dock any of the cofactors in a sequence. And then for a one-off time, we want to find the residues that we want to fix in our sequence for when we're redesigning the protein, because it's an iterative process. So we're just going to keep one section of it fixed the whole time. And hopefully that should conserve the functionality whilst also giving ligand MPNN the room to be able to improve our protein. And then in this section here, we'd run a bunch of ligand MPNs to get like thousands of sequences usually, and then we'll then rank them. We'll take the top ones that we think are good enough, and then we'll get the 3D structure from the sequences in order to check that we think, okay, this is past the test. We're happy with this sequence. So then after this, we repeat the cycle again and again until we start to plateau with ligand MPNN, and we're no longer really getting new results which are like improving any further. Okay, so first of all, the 3D structure. So why do we want the 3D structure of our protein? Well, the 3D structure of the protein corresponds to how the protein will fold. So that essentially corresponds to the functionality of the protein. So by getting the 3D structure from the sequence, we're able to tell the functionality of our protein, which is quite important for when we're designing, because if we get a completely different structure, then we can go, oh, right, we're clearly not going to get the same functionality here. So I'm no longer redesigning now. It's starting to go into, I've created like a newish thing, which is not the aim for what we're trying to do in this walkthrough. Why do we want to dock the cofactors? This is probably going to be a bit important further on, but some of the redesign tools, we want to try and figure out the important areas in our protein. So, for example, the active site is an extremely important area for the example that we're going to do, because we're going to do PAFA, which is an enzyme. So we need to know where the active site is in PAFA. So if we dock where the zinc ions are and we dock a ligand in, then we're able to figure out where the different residues are around there. And then when we tell ligand MPNN, okay, these are the residues that we want to fix, it should hopefully mean that we're going to be able to maintain the functionality. So what we're going to do is we're going to go into AlphaFold first. So we're going to get our sequence from benchling. So we just click on our protein in benchling, and then you can right click and press copy, and we want the amino acids. So that's on our clipboard now. And we're just going to use AlphaFold for this example, and we're going to do docking with AlphaFold as well. So we have our job here. So what we do is we paste in the sequence of our protein, and then to dock different cofactors in, we just add an entity, and then in molecule type, you can change this to be whatever cofactor you like to dock. So we want a ligand and we want the 2 zinc ions. So we'll add in the 2 zinc ions. So you can just change the copies there. Oh, sorry. And then we also want our ligand as well. And we'll just use AMP for this example. And then after this, you'd say, continue and preview job. You give the job a sense of name. So you can just put in whatever you like. And you don't need to worry about the seed. And then you just press confirm and submit. And then that would be it. So we have an example already of one of these jobs that we've run. So this is our output from if we'd chosen to run the job and then waited for it, and then we go, oh, here we are. It's quite quick on our fault server, the wait time. And as you can see, we've got really good scores. Well, mostly really good scores for our model. And then our error rate is also extremely low as well. As Felix was saying earlier, the lower the error, the better it is. So then from this point on, you download a zip, which would basically have all the models from AlphaFold, and then that'll be only keep it. So we'll just go back to the presentation. So vaguely set the scores. So then on to fixing residues. So we want to figure out the residues that we want to fix, because we've got the 3D structure in our cycle. We've docked the cofactors that we want. So now we need to figure out which parts do we want to fix. So why would we fix the residues? Well, I think I've already said this before, but essentially we want to fix the residues so then we can try and keep the functionality. So we're going to use two tools for this. We're going to use Conserve and we're going to use ChimeraX. So Conserve, all you have to do for Conserve is upload a .pdb of your protein. So this is a thing with AlphaFold as well, actually. So we'll briefly do this in ChimeraX. So I'll just get rid of that. Sorry. So if we have a .civ, AlphaFold gives us .civs, which is just another way, just another file type for 3D structures of proteins. But if you ever want to change it from a .civ to a .pdb, all you have to do is say save, put in the name of your protein. So we'll just say something like random, and then .pdb, and then that'll now save your proteins as .pdb. So then for using other tools, at the moment it seems loads of other tools use .pdb. I think that's just the general format. So this is just a quick, easy way to get it into that format. And just with ChimeraX as well, if you want to know where you've actually saved the file, you can use pwd, and then it'll tell you where it is on your computer, which is quite useful. So back to Conserve. So you've got your .pdb now of your protein that you want to find the most conserved residues. So you just upload the PDB there. If you've been following through with the walkthrough, when it says select chain identifier, in the .pdb file, it puts all the different elements of the model, so the ligand, the zinc, and the protein on different chains. So if you've just done it the same way as this, whatever's first will be A, B, C, D, E, as you know, for as many cofactors as you add. So you'll always probably pick A if you're going to put the protein in first. And then all you need to do then is type in the name of the job and your email address. And eventually you'll get an email back from Conserve, and you will be able to see the results here. But this is probably not the easiest way to scrape out the data that you want. So what you do is you download all the files, you get a little zip, and we have a Python script on a Colab, which basically just means you can upload a file here, and then all you have to do is change the name of your specific file name. So I'll just show you what it looks like, the Conserve results. So the Conserve folder will usually have all these files in it, and it'll just have the same name as your .pdb. And this text file here has the best data for you to use, although it's not in the easiest format to play around with. So you sort of want to get into like an Excel document. And the main thing you want to take away from it is the highest scoring things and the position on the chain that it belongs to pass into ligand MPNN, because that's how it fixes residues. So all you do here is you put in the file name of that TXT that you get from Conserve. You put in the file name that you want, for which you just copy from that file. It would have the chain and just the number, so that relates to the position of that residue in the sequence, and then the percentage that you want to keep conserved. So for our project at the moment, we've looked at 30%, 50%, and 70%. There's no like defined percentage to conserve when redesigning, because it'll vary based on the protein that you're trying to improve. But from some of the Baker stuff, it was like 50%. You tend to still have the same functionality, but you also get the most amount of improvements. So 50% seems to be like a safe option going around there. But if you went 30%, turns out it worked. You're probably going to get more improvements there, but it just varies. So yeah, you pop in your file here, get your data out about how to which residues to consider, and then obviously there's Chimera X as well. So you have your webinar. So you have the .pdb file now of the protein that you got from AlphaFold just a moment ago, and you have the different cofactors which you docked into it. So you want to find out the residues around it. So with AlphaFold, with Chimera X, you can select different sections. You want to select a set amount of angstroms away from these cofactors, and about 5 is a good amount to select away to figure out the residues that you want to fix. So in order to do this with Chimera X, you just say select, and then if we're going to do it for the ligand, we'd say ligand, and then @ is for angstrom, and then less than 5, and then this would select everything less than 5 angstroms away. And then in actions, you can use label, and you can label the residues. And I'd say chain code number is a pretty nice one to go with. And then this will let you be able to see all the residues that you've selected. So it'll even show you all the atoms as well, so like zinc, and it'll show you the amp in it as well. And there's also another command which is quite useful for letting you see what you selected. It repeats itself a bit, but then you can write those down, or you can put them into the file when it's time to run the script to use ligand NPN. So carrying on from that, it is probably time to use ligand NPN. So we'll quickly go through a script for ligand NPN, because that would be quite useful. So here, I'll just use size one, then it's a bit small. There you go. So in the ligand NPN, you'll have this one Python file in it called run.py, and that's basically the main program for ligand NPN. And in order to use ligand NPN, you'll want to select the model type. So you'd set different switches. So in the Darkness ligand NPN thing, there's a bunch of switches that you'd use to try and use ligand NPN. So the switches are basically just options. So the dash, the name of whatever it is the option that you want to select when you're running your specific model. So for this example, we're going to use the model type to be ligand NPN because we're going to put ligands into it. You have the seed, which is basically where it would start off in the neural network. That doesn't really matter where you set that, but just use a set number. You've got the PDB underscore path, which is basically where you're putting your .pdb file that you just got from AlphaFold. So that's the where the structure is of the protein that you want to improve. So if we look in my files, and in my ligand NPN folder, there's an inputs folder there, and then we have the webinar underscore example there. So that's all we're writing for that PDB path thing. And then we also have an output folder. So that's where we're going to get all our results from ligand NPN. So that's where you're going to find all the sequences that you have improved. And as you can see, this has already been run before. So I now have an outputs folder, which has all the results here. I'll go through that in a moment. And then probably the most important section for redesigning is the -- fixed residues section. And this is just all the residues that you don't want to have changed using ligand NPN. There's other switches which are available which can do the reverse. So it's like, please do change these ones. But for this example, we're not using that. And it's also important to keep your cofactors fixed as well. So B1, C1, D1 relate to the ligand and the two zinc ions, but the rest of this is all related to the protein. So it's just the position of the residues in the protein, and then stating which chain it belongs to is how we fix the residues. And then finally, it's the number of batches. And this basically just corresponds to the sequences, the amount of new improved sequences that you're going to get out. So in order to run this script, you just say, you just run the path.ps1. And then hopefully this won't take very long to see it happen. I believe. Essentially, it'll run and you'll get something that looks a bit like this, and then that means everything's worked and everything's okay. But I've already done this, so we have an outputs folder here, and this has the improved sequences in it. And I don't know if the two second. Yeah. Okay. So yeah, we're going to check through the new sequences now. So these are our new sequences. So the first line here basically tells you how your ligand MPNN program ran. So it gives you all the information about how it ran. The first sequence is the original sequence that you've improved from, and the rest below are all your new improved sequences. So you get a load of data here. So you have overall confidence, ligand confidence, and then sequence recovery. And they're not the most useful metrics for going, oh, has this really worked well? It's kind of like, it's not the best way to go about it. But in terms of our redesign cycle, we decided with going through it with higher ligand confidence means it'll probably be a better result. So more likely that it will improve incorrectly. So for example, on this set of 10, number 18 had the highest ligand confidence. So what we do now is with our new improved sequence, we go back to, well, what do we do with the outputs? We go back to, so how would we check if the new improved sequence is good? So we go back to AlphaFold now. Here. And then we put our new sequence back into AlphaFold, and then we get the new structure as well. And as you can see, it looks vaguely similar. So the way to check that it actually is about right is if we go into Chimera X again, we'll just unselect things and unlabel things. So we have our original protein here from before. And then if we open our improved one, if we use the command matchmaker, which is just MMAker, and we go #2 to #1, that's just the IDs of the models. You can see that we've got basically an extremely similar structure, which is very, very promising. Obviously, there's some bits which are, a little bit different. So, like some of the strands at the ends have gone off a bit, but the core, the central part of our cafe has stayed the same, which is really quite good. So we checked it. Oh, and the other good way to check it is if it's less than 1 angstrom across all pairs when you've done the when finding the root mean square deviation between the two models. If it's less than 1, you'd go, okay, this is a good result. I'm happy with this, and then I'll go on to the next stage of the cycle, or I'm ready to finish. But if it's above, then you can go, oh, this might just be a little too off. My structure's just a bit off now, and I'll leave it. So we've checked the sequences, we've compared them, and we've gone through what to do if it fails. So next steps. That's it. We've improved our protein. It's brilliant, but not yet. So what we should do is we should go through a cycle where we take this new improved version, basically do the same thing again, and once the ligand confidence levels have got to about their best, or they've plateaued off, or it's like the next iteration has just got worse, you can go, okay, I'll go back to my previous iteration, I'm just going to take this as my best one. And then after that, obviously, because it's only models and estimations, it's time to send off your G-blocks to see if it actually works. And that's it. That's your walkthrough. on how to redesign proteins.
Do you have quick few minutes for questions? Anyone ask anything in
I'll have a look.
chat? Chat, is this real?
No. I don't think so. Yeah.
Well, you about to say a bunch of exclusives.
I you're going to make the recordings available.
Yeah.
I think, yeah, the point of sort of this is that I generally really like it when you do that. It's like a guide for helping people.
I suppose, are there any questions?
I actually have a question that I've not considered.
Yeah.
In MPNN, does it automatically assign the first amino acid to be methionine?
No.
You have to tell it to.
You have to tell it to.
So when we fixed our residues, have we fixed residue 1 to always be methionine?
Yeah, I did fix residue. Well, yeah, I did after it. I did one result and I was like, I'm getting A's as my number 1. This is not correct. But
So that is something you will have to.
I suppose it's like if you were always going to assume something methionine, it's always got to go first. If you're going to redesign it, if you're going to redesign it, then you might just like skip out that because you know it's going to be. I don't know. Maybe it's better to ask Felix.
No, I think I just don't include the design anywhere around.
Yeah,
So it's just like, is that one?
Oh yeah.
because it's always going to be there. So there's no point redesigning.
Does there ever any other residues that count as a first residue in any weird exception?
Sometimes they're like very transformation.
Yeah. I mean. Okay, well, question, we can keep taking questions sort of throughout the next bit, but I think the last kind of portion of the talk, we're going to hand over to Professor Williamson. He's going to tell us about why and how far can we take these tools and perhaps what are some of the limitations of using AI in structural prediction.
Yep, thank you. So if you're all getting tired of all these presentations, this should take 10-15 minutes, so stay with it. Okay, so from the talks you've heard so far, hopefully you've got the feeling, wow, this is really great, I can do this. This is so easy. I'm actually not really going to say no, it isn't, and no, it doesn't work, because actually basically I'm going to say it does work. You just have to know what you're doing. So, and it really does work. So I always used to give a lecture to the very first lecture that our undergraduates ever had at Sheffield, which was basically to say, we don't know everything yet. There's lots of things that we really don't know. And one of the big things that we didn't know at the time was, can you predict a protein structure from a sequence? Because we were really useless at it. And then AlphaFold came along and it solved the problem. It really has. And so their biggest development in structural biology in the last 20 years, at least, I'm sure they're going to get a Nobel Prize at some point. It was actually first presented at a CASP meeting in 2018. So CASP happens every 2 years. It's a structure prediction meeting. Everyone comes along with their new computer programs and shows how well they do. And it blew everybody away in 2018, and then they completely went away and rewrote it to make AlphaFold 2, which they presented at the next CASP meeting in 2020, and that's just a picture of the results from 2020. So the CASP results are the first bars, the kind of dark purple ones. Everybody else is the next ones, and you can just see how much better AlphaFold is than everybody else. Interestingly, actually, the second, third, and fourth, I think, are from the Baker Lab. So this is the MPSN thing, and Rosetta. So they're pretty good too, but at the time, at least, nowhere near as good as AlphaFold. So AlphaFold really is spectacularly good. It really genuinely is. Don't let me give you the impression it doesn't work, because it is amazing. And since then, AlphaFold 3 has just come out, and my understanding, although you can tell me if I'm wrong, is it's for predicting a single protein chain is fairly similar, but for predicting complexes and ligand-bound structures is really a lot better, and it's certainly very easy to use. So it's getting better all the time. Okay. Sorry, lots of words in my slides. AlphaFold is an AI, artificial intelligence program. A very few words about what an AI program is. So an AI program, artificial intelligence, is simulating the way our brain works. So it's a neural network, meaning basically what you do is you have an initial layer, which is the factors that you think might be important. And then there's a second layer, which is some combination of the things on the first layer. And the computer is working out what the weights are, so how to combine those. And then you can have as many intermediate layers as you like. And then there's a final layer at the end, which is the output. So essentially, you kind of give it a structure of a brain, and then it fills in all the gaps and it learns how to solve the problem. So what you're doing is you're training it. And training, as I understand it, means essentially you give it a load of input, so for example, protein sequences. You give it a load of output, that is the structures that those protein sequences should adopt. Typically, the X-ray structures correspond to those sequences. And you train it by saying, well, here you are. It's an input, it's an output. This is how it, what we want you to get. You go away and learn how to do that so you can do it with new sequences. So that's the training phase. And it is basically was trained on crystal structures, and I'll come back to that in a second. CAST 13 was obviously the 13th CAST meeting. There have been lots of, it happens every 2 years, it's been going on for a while. There are lots and lots of programs out there that try and predict structures from sequences. Essentially, all of them before AlphaFold, what they were trying to do was calculate the forces between atoms and groups and amino acids, and how those forces work together to fold the structure up into the structural plans. So essentially, they were trying to simulate the way a protein folds up in solution. AlphaFold does not do that, or at least basically AlphaFold doesn't do that. What AlphaFold does actually is it looks at protein sequences. So you give the sequence of your protein, it goes away and looks in the database and finds all the other proteins that have similar looking sequences, and it compares them. What it's actually looking for, as I've put up there, is covariance, by which I mean that if your sequence has an amino acid somewhere, say a leucine at one position, and every time I come across another sequence that has something else not leucine there, there's another place somewhere else in the sequence that was a phenylalanine originally, and that's changed as well, particularly if they're changing in some kind of concerted way. So, you know, if a leucine changes to a phe, this phe changes to a leucine. Not always nearly as simple as that, but that kind of idea that if this amino acid changes, that one also changes. They have a high covariance. They're very likely to be connected in some way. And the obvious way they're connected is that they're next to each other in the structure. So basically, AlphaFold, very simplistically, is predicting the structure by mapping things that have high covariance and saying they should be close together. So it's not using forces at all. It does a little bit now, but I think still it's basically using sequences. So it's actually doing in a not at all an intuitive way, not the way you think that protein a structure would get calculated. And it's amazing it works as well as it does, and it really does work amazingly well. Two interesting consequences of that being the way that AlphaFold works. One is because it's taking known sequences from all organisms and you know, different strains of different organisms and matching them. If what you're trying to calculate is something which is not a natural protein, so if it's a mutant, or if it's just a complete de novo sequence, then it's got nothing much to compare it to, so it doesn't do so well. That was certainly true for AlphaFold and AlphaFold 2. I think it's not so true of AlphaFold 3, but I would say you probably still want to be a bit more careful if you're giving it a sequence which is not a known sequence. Second one, I think, is an interesting question and not quite as obvious. The training that AlphaFold got was to say, well, here's a sequence, here's the crystal structure of that sequence, and that's the training. So it's trained to produce something that looks like a crystal structure, because that's the data it had that it was given. Is that what you want? So if you're trying to calculate the structure of a protein, do you actually want the crystal structure? Is the crystal structure the best representation of what that protein looks like? So I'm an NMR person. NMR looks at things in solution. And in a sense, if you're trying to model the structure of protein, you don't actually want what's in the crystal. You want what's sitting in your cell, in your solution. So you'd rather have it predicting something that's in solution rather than something in a crystal. Does that matter? It matters if the crystal structure is wrong. So interesting question, which has been quite a hard question to answer actually, is are crystal structures actually not quite right because they're in a crystal? Does being in a crystal distort the structure in a way that matters? So I've got a couple of slides kind of thinking about that. So here's first one. We looked at this question. So basically, we developed a computer program. Sorry about that, something funny happened to my layout there. We developed a computer program that basically looks at structures and calculates the local rigidity of the structure. It's a graph theory thing. And then it also takes NMR data for that protein in solution. And you can work out the local rigidity from the NMR data. So it's calculating the rigidity of the structure, calculating the rigidity as measured by NMR, and saying, are they the same? If they're the same, then your solution is, your structure is correct. If they're different, your structure is wrong. And that picture at the bottom there is basically comparing NMR structures to crystal structures. The crystal structures are in blue, and they've got lots of things at nearly 100%. The NMR structures are in orangey brown, and they're about 50%. And basically what that's saying is the X-ray structures are a lot better. So the X-ray structures have a better rigidity that matches what you see in solution, despite the fact that the crystal structure was determined in crystal, and the NMR structure was determined in solution. So from that measurement, you'd say, does it matter that it's a crystal structure? No, because crystal structures are actually very close to what the genuine solution structure looks like. So that's the crystal structures. And then on the next slide, we look at AlphaFold. So we did the same kind of thing with AlphaFold, saying if you look at the AlphaFold structures and compare those to rigidity of what we know about the protein actually in solution, is AlphaFold right? And the answer is yes, it is. So essentially what that graph at the top is saying is that the AlphaFold structures are about 28% better than the corresponding NMR structures on average. There are some that are worse. But on the whole, the AlphaFold structures are much better than the NMR structures. Again, even though AlphaFold is basically trying to calculate what the crystal structure would look like. So the AlphaFold is basically trying to model a solution, a crystal structure, not a solution structure. That's mainly because NMR structures actually have a very good hydrogen bond. Different topic altogether. There were a few structures for which the NMR structure was clearly better. I'm showing you one at the bottom there. So the one in blue is the AlphaFold structure, which was basically an alpha helix. And the NMR structure said, no, it's not actually an alpha helix, it's two alpha helices with a kink in the middle. You look at the NMR data and it's absolutely clear that there really genuinely is a kink in the middle there. So it actually does look like the NMR structure. AlphaFold, if you like, got it wrong. But AlphaFold is trying to predict a crystal structure. Now, if something's going to crystallize, and I think Felix said this, like somebody said this, or somebody said this, if something's going to crystallize, actually it has to be regular and it has to fold the same way in all the unit cells. So if you like, a crystal structure is in some ways a bit kind of artificially over rigid. So if AlphaFold makes a mistake, usually the mistake it makes is to make the structure a little bit more rigid than it probably is in solution. And the way you know, and people have talked about this before, I'll show you a bit more about this in a minute, is one of the things that AlphaFold reports is a thing called the PLDDT score, which is basically how much AlphaFold trusts the result. And the PLDDT score at the kink in the AF2 structure was actually pretty rubbish. So if you like, AlphaFold did know that it wasn't a very reliable structure. And anyone who knew what they were doing should have looked at the AlphaFold structure and said, don't believe that. So actually, are they different? No, not really. And AlphaFold basically was correct, I would say. I thought I'd give you another example. This is a protein I'm working on at the moment. This is a protein trimer. So basically what I'm trying to do here is calculate what the trimer looks like. It's a complex of 3 proteins, 1, 2, and 3 there. Again, the thing on the left is the 3 proteins as a complex coloured by the PLDDT score. You've seen these before. So when it's a dark blue, the PLDDT is more than 90, really confident. Where the PLDDT score is yellow or orange, it's fairly unreliable. And you can actually see here, so most of this protein is blue, so it's pretty confident. Most of this protein is blue. Most of this protein is blue, so it's actually fairly confident about the structures of the 3 monomers. But actually, if you look here, there's a whole bunch of stuff which is yellow and purple, orange, sorry, in the interface between protein 1 and protein 2. So it's saying, I know what protein 1 looks like, I know what protein 2 looks like, I know what protein 3 looks like, but I really don't know much at all about what the interface between protein 1 and protein 2 looks like. And again, you've seen this kind of thing before. This is the PAE score. And it's again saying, okay, I know what protein 1 looks like. I'm pretty confident about that. I know what 2 looks like. I know what 3 looks like. Again, where 1 is in relation to 2 and 3, I have absolutely no idea. So it's just a complete guess as to where that should be. And it's actually wrong. Okay. That's basically everything I wanted to say. So what lessons am I trying to draw from that? AlphaFold is terrific. It's a wonderful tool. It really does work. But like any tool, you've got to use it properly. You've got to know what it's doing. You've got to know how to interpret it. You know, when I look at the AlphaFold result, do I believe it? Well, actually, if AlphaFold is saying, no, I don't really think this is any good, don't believe it. So you've got to know how to use what you're doing. That's obviously true of almost anything, but I'd say it's particularly true with things like AlphaFold. Interesting point, actually. AlphaFold, it's an AI program. An AI program, you start off by, when you're programming it, you give it your initial layer, and then you tell it to kind of construct all the intermediate layers and work out all the weights. So you have really no idea what it's doing. And I think this is true for AlphaFold as well. I don't think even the people who wrote it really know what it's doing there in the middle. It is a black box. So no, it's like your brain. Your brain is not obviously an artificial intelligence, it's intelligence. But the example I like to give, my apologies to those of you who are not English speakers, this probably works better if you're not an English speaker. If I were to say to you, oh, look over there, there's a black big dog, you would say, this guy's not English, it should be a big black dog. And then I say to you, why is it right to say it's a big black dog, but not to say it's a black big dog? What's wrong with saying it's a black big dog? I think if you learn English as a foreign language, they do tell you that there is an order in which adjectives come. But, you know, I didn't know that.
Yeah.
So what I'm trying to say is, in the same way that I don't know why I say the things I do, I just know it's right. I think the same is true with AI programs. They work. Even the people who program them don't actually know how they work. And we do have to treat it with a little bit of caution, because we don't really know what it's doing. So my take-home summary, AI is fantastic, AlphaFold is fantastic. It's an absolutely brilliant way of getting ideas. But as you heard from Tom, you've got the ideas, you've got to then go away and actually make it express the proteins. It actually works. I think that's me done. Thank you.
So, yeah.
Is there any questions in the chat?
I'll have a quick look.
No, I have a question that might actually be relevant to people, directed at both you guys. I think it would be useful. What is the best way of interpreting when AlphaFold has a low confidence in a particular part of structure? Because sometimes it's my understanding that if you have a protein that requires some kind of hinge, that it's meant to be good, flopping about as part of its function in some way, is it reliable to interpret the kind of hinge bit as when it says that it's not confident? Is that actually going, oh, cool, it knows that it's not confident there, therefore it kind of knows that it's meant to be flopping around. That's good for your, because you can imagine if you needed it to be hinging, and actually the structure between the two units that were hinging, that's definitely not word.
I'll let you answer. I mean, my understanding is actually a low PLDDT score is one of the best predictors of local mobility.
Right.
So, yes.
But it's also a mixture, I think. Like, it definitely is, but then it's also there'll be situations where it's like alpha just failed.
Yeah,
So it's like you have to look at it and go, does this look sensible to move in that way? There's something I was going to mention when we talk about covariance. Something that so someone came out. So alpha, obviously, if you select a single seed, it only outputs one conformation of a structure. That's very annoying for things like transporters. And then Group came out with a project where they took a glucose transporter and they only got one conformation. They ran a covariance evolution study on it, found the covariant residues, and then used molecular dynamics to solve what the other conformation should be. And there are tools online that you can actually do this covariance analysis. We have something that's based on like the multi-family cellular transporter domain that's like in some photosynthetic organisms. And alcohol always outputs it with like open out sort of way. And using Rosetta, you can like do a relax sort of thing. It's like time independent molecular dynamics using some sort of physics-based thing. So I was going to say, you showed your alpha folds versus NMR structure.
Yeah.
I wonder if you put that alpha fold structure into Rosetta, that region, the alpha fold is made of helix. I think that will probably become a term if you put it in Rosetta. Yeah. So yeah. So one of the structures I got out of alpha fold, I did the covariance analysis, didn't do any restraints in Rosetta, and I just let it relax it. It found the other covariant partners. So when I did the covariance analysis, I said, yeah, all these should be close to each other. They look close to each other in the alpha structure. I said, these guys up here also should be close to each other. And then when I Rosetta relaxed it, it did that. And so it's like, there's obviously other information in these structures that you can tease out with other tools. And I think alpha fold is maybe just not the last place to stop.
It's interesting how even with that, you have to go back to some physics-based tools. So I wonder if like a future like AlphaFold form a cool tool would be to do some of those molecular dynamics simulations, and for things that do have a predicted change, it spits out animation rather than like statics.
It's an interesting question, and I don't really understand enough about the physics to know this. So, yeah, I mean, molecular dynamics is just predicting how things move. And the problem is that proteins don't move in any kind of smooth way, if you like.
Yeah.
So it wouldn't actually do a helix bending like that. It would just jiggle it back sometimes.
Yeah.
There is a thing called, which I think is kind of what Felix is talking about, a thing called normal mode analysis or elastic network modeling, which is basically trying to say what are the bits of movement that go together most, or what the lowest frequency normal modes actually. And that is exactly, and that gives you, if you've ever seen pictures of proteins kind of waving about, that's normal mode analysis, and that's actually very powerful. That's cool. It's quite quick too, much quicker than molecular dynamics.
We do have a question. Why couldn't an AI tool be built on both NMR and crystal structure?
I was also going to mention that as well. Do you
It could. Yes, it could. I think so. I've been talking to various people about why AlphaFold works so well and why it's taken this long to come out. And basically what they said is essentially it's just because computers have got an awful lot faster and there are an awful lot more crystal structures. So I'd say the reason it works so well is it was trained on lots and lots of crystal structures, and crystal structures are good. There are not so many NMR structures, there's about 10% NMR structures compared to crystal structures, and they're less good. So you could do it, but it wouldn't work so well.
I wonder though if, because it seems that like NMR does point out things that perhaps crystal structures would not. Because like you say, crystal structure is definitely like an artificial, I've watched the protein in this shape just to get a sense of what it's doing.
Yes.
Whereas I didn't realise that NMR was all based in solution, so it's much more dynamic. I think
Well,
it seems like
you can do solid state NMR, but it's a different thing.
I see. Well, I don't know. I still, from a kind of just intuitive point of view, I wonder if it's still
actually, if you look at a protein crystal, I've forgotten what the figure is. It's over 50, but I think it's about 60–70% of a protein crystal is water. So actually, although it's in a crystal, it's not as if it can't move.
I see, right, okay.
It can move quite a lot. So you do get what they call crystal packing effects. So yes, you do. And occasionally things are a bit weird because they're forced to because it's next to another one, but it doesn't actually seem to make much difference.
Oh, interesting. Cool. Anything else from the chat?
Nothing else from the chat? I think that-
Anyone in here got any final questions? Lovely. Thank you. Thank you for joining us today. Thank you very much to our guest speakers, Felix and Professor Williamson. I hope this guide is helpful, and a written guide that summarizes our use of some of the AI tools will be ready in the next couple of weeks.
Yeah, I think it should be ready reasonably soon.
Yeah. It's the end of my time. Thank you very much.
Thanks, guys.
Now we've got to pause until, like BBC News, share the screen.